Through modern medicine, we've made incredible strides in improving global health. Yet when someone is diagnosed with a condition like hypertension, diabetes, or even depression, they're given a drug, asked to try it out, and then come back if it doesn't work. We rely on this trial and error approach because we're all uniquely different, and our differences come from the very building blocks of life, our genes. Every cell in your body contains a complete set of instructions in the form of genes, and these genes dictate everything, from how you develop to how your body responds to medication. But this diversity also means that no single drug will work perfectly for everyone. It's like trying to design a universal key for every lock. It's simply not feasible. Now imagine if you could sequence your DNA and create a digital replica of your cells inside a computer, sort of like your digital twin. You could then use this computer program to virtually test your body's reaction to different medication without any risk to you. And I know this might sound like science fiction, but thanks to recent advancements in AI and the large amount of genetic data that we have today, we're beginning to see early versions of human virtual cell models. Unfortunately, most of these models treat cells as a mysterious black box. So they might tell us that a gene change can improve a cell's condition, but they don't reveal how. And without understanding the underlying mechanisms, it's almost impossible to improve these models unless, of course, we incorporate more genetic data, which is costly and time-consuming. That's why I'm developing a glass box model, where you can peer inside and see exactly what's happening. Specifically, my model creates a detailed map of hidden genetic conversations, showing exactly which genes are communicating, what messages they're sending, and how these signals regulate everything inside a cell. Here's an example of a genetic map that my model has discovered. And as you look at it, I hope that you appreciate the complexity of human gene regulation, but also realize that uncovering such an interpretable model is not that easy. And that's why I'm using a tool inspired by nature itself, an evolutionary algorithm. Just as natural selection refines species over generations, my algorithm systematically evolves potential models by 1, slightly modifying them, just like in mutations, and 2, by blending the best ones together, similar to reproduction. And this goes on until we find models that can accurately simulate observed cell behaviors. This novel approach can make gene regulation more transparent, and one day move us beyond the frustrating trial and error approach plaguing modern medicine. And instead of blindly trying different drugs on patients and hoping for the best, we can simulate their unique biology in a virtual cell and predict the most effective treatment from the start. These virtual cells will also deepen our understanding of how cells work normally and how they might progress to disease when something goes wrong. And while there's still much work to be done, these advances bring us closer to a future where medicine is no longer a guessing game, but a precise science tailored to each individual. Thank you.